Transcription occurs in the nucleus. Translation occurs where? Because we already did this. What organelle is responsible for making proteins? Ribosomes. So translation occurs at the ribosome. And we call it translation because the message is now being changed from a nucleotide message to a polypeptide message or a protein message. So we're going from one language to another, for lack of a better word. So we call it the translation. And actually, Tim, to you, the answer to that question is going to come in about 17 minutes, not 7. Maybe the drawing first. First thing that happens is that messenger RNA, remember that was created in the nucleus, leaves the nucleus and goes out to the ribosome. The ribosome itself is made up of a large and a small subunit, and the main ingredient in ribosomes is ribosomal RNA. So we got messenger RNA that carries the message. We got ribosomal RNA. We're going to find out it's the thing that actually makes the new protein. So in a lot of ways, you can think of this as a construction project. You know, DNA is the architect, makes the blueprints, gets what you want. Make a copy of those blueprints, that's the message. The message is going out to the work site saying, make this. At the work site, we have the ribosomal RNA. It's what's going to build whatever it is the message says to build. So when we get to the ribosome is going to attach to that messenger RNA. So now if you think about it, we've got the workers, we've got the plans. What are we missing? Missing materials. Materials are going to be transferred to this work site by tRNA, or transfer RNA. It's going to bring the amino acids to the ribosome to assemble them into new protein. Now, it turns out that there are really two important sides to the transfer RNA. Transfer RNAs have an amino acid at one end and something called an anticodon at the other end. And the other end, the anticodon, is complementary to the codon. It's how the transfer RNA knows where to bring the amino acid. Because of this complementary nature between the anticodon and the codon, we're going to know where to line up the amino acids. And once these amino acids are next to each other, and it's really the ribosome that's going to hold them together, once the amino acids are next to each other, then those amino acids are going to form a bond. And what chemical process is going to allow the amino acids to bond together?
Covalent bond?
Say again? Sort of, but what do we call the reaction that makes us stick things together? Via the removal of,
Dehydration synthesis.
No, go ahead.
Ionic bond? Double one bond?
Well, they are going to be part of those bonds, but it is going to be the dehydration synthesis. We're going to remove water, bring these things together. So the amino acids are going to form these peptide bonds by dehydration synthesis and keep growing into this polypeptide chain. And then as those bonds occur, the ribosomes are going to continue to move along that strand. So that ribosome is going to move along, we're going to bond a new amino acid, ribosome moves down. Bond the next amino acid, ribosome moves down. And it's going to continue going all along that messenger RNA chain. And again, a lot of this will be easier to visualize once we do the diagram, which we're going to do after we finish one more slide. And we hopefully will see it. There is a picture of it in your book. There are animations of it online. If moving pictures make you feel better. But we'll also have a sketch of it so we understand what's going on. As it turns out, there are some important codons. First thing we need to know, and this is what the exercise we just did on the board, each codon only codes for one amino acid. So UUA always codes for leucine. It doesn't code for anything else. Because you can't have a code if the code word codes for a bunch of different things. That wouldn't make any sense. One of the challenges we have with protein synthesis is the RNA that leaves the nucleus has a lot more to it than just the code that we want. What leaves the nucleus might be 200,000 or 300,000 bases long. We might only need 10,000 of those bases. So one of the more important codons is AUG. AUG is the initiator codon. AUG means start here. So when that codon gets to the ribosome, it's going to ignore everything in that chain until it gets to AUG. So if AUG means start, what amino acid is always going to be the first amino acid in every protein ever created anywhere? No, AUG is a codon, but what is that code for? It codes for methionine. That's why you'll notice if you look at where AUG is on your codon chart, it is the start codon, but it also codes for methionine, which makes methionine, at least in the primary structure, the first amino acid in every protein ever created everywhere. Well, if we've got 100,000 base pairs and we only need 10,000, what's almost as important as knowing where to start? Where to end. And it turns out there are 3 stop codons or terminated codons. UAA, UAG, UGA.